We're very excited today to have with us, Melis Akguen. She's a PhD candidate and in fact just submitted her thesis yesterday, she told me. So congratulations with that. That's fantastic. A little bit about her. The title for her talk today is Resolving Human Alpha versus Beta Cell Fate Allocation for the Generation of Stem Cell-Derived Islets. And just a little bit about her. She does already have her master's. Again, she's a PhD candidate at Helmholtz Zentrum München Institute of Diabetes and Regeneration Research, Munich Technical University in Munich, Germany. And she just was awarded the best short talk at the Gordon Research Conference in Italy, and also the DZD Outstanding Young Investigator Award for 2024. So congratulations on both those counts. It's pretty impressive. And really thank you for joining us today, Melis. We're really interested to hear about more about your work. I'm going to put something in the chat here for everyone to please ask questions, to encourage them to ask questions here during the talk. And we will interrupt as needed, and then we'll also take questions at the end of the talk. So thanks so much for joining us, and take it away.
Oh, thank you. Thank you for the kind introduction, also the invitation. I'm a senior PhD student, just submit the thesis yesterday. So in the Helmholtz Diabetes and Regeneration Research Center in Heiko Lickert's group. So today I would be happy to share my PhD work with you. But before diving in the alpha versus beta cell fate decision in vitro human model, I'd like to give a little brief to explain how this idea evolves. So as we all know, the pancreas regulates our glucose homeostasis in our body, mainly secreting two hormones, glucagon from alpha cell and insulin from beta cell. And the diabetes develops due to the beta cell loss or loss of function. Therefore, in our field, the beta cells are prime target for cell replacement therapy. So they have in many different labs shown in different model organisms, the inter or intra tissues cells can transdifferentiate to beta cell, but considering the close physiological relationship of the alpha and beta cell, as well as the big transcriptome overlap between them, so alpha to beta transdifferentiation draws much more attention for the beta cell replacement therapy. And for the sake of today's talk, I just want to highlight one of the studies. Even the one single gene manipulation, in our case, the PAX4 overexpression, has been shown to sufficiently trigger this system. So another way of the cell replacement therapy, in our lab, also in a different lab, the idea is appealing to generate the insulin producing beta cell in a dish. Now we are taking advantage of the using pluripotent stem cell, and we are able to generate differentiate them towards the insulin producing beta cell using a directed in vitro differentiation protocols. Now I think more than 30 different beta cell protocol is published, and there's all the protocols aim to recapitulate pancreas development based on the mouse findings. Therefore, we also check the efficiency and quality of the generated cells using the stage-specific markers. Here, we aim to generate first differentiate definitive endoderm, then primitive gut tube, then pancreatic progenitors, endocrine progenitor, and finally hormone-producing mature endocrine cell or endocrine-like cell. But still, we are seeking for a lot of improvement in different topic in different problems in this differentiation protocol. But today, I would like to highlight one of them. It's differentiation efficiency, maybe the most important one, because in a dish, we are able to generate relatively homogeneous population till pancreatic progenitor stage. And then we generate more or less 70% pancreatic progenitors expressing both progenitor markers, PDX and NKX 6.1. However, we face the heterogeneity in endocrine progenitor stage. And this is the stage where the endocrine induction occurs, and these progenitors further differentiate, further decide their fate. Mostly it's alpha or beta cell fate. And this heterogeneity further then yield relatively low number of the hormone producing cell. This is one of the reasons we want to tackle this question, and we tackle these problems, and we want to understand what governs this endocrine induction and also alpha versus beta cell fate decision in this differentiation protocol. But first, we need to go back to the proposed mechanism for this, and it's on the mouse endocrinogenesis. So what we know, it is very well described 20 years ago, so maybe more. So the endocrine precursors expressing NGN3, which is a master endocrine precursor marker, can further differentiate the pancreatic endocrine cell, mostly alpha cell or beta cell. And there are two main transcription factors govern this process, ARCs and POX4. They have been shown in the mouse knockout studies. First, they generate the POX4 knockout mice, and this mice had lack of all the beta cells, concurrently increased number of the alpha cell. Another lab also generate the ARCs knockout mice, and this mice, they shown that it's lack of all alpha cell and concurrently increased beta cell number. And the same Colombat lab also generate B allelic mutation, meaning that they knock out the ARCs and POX4, and they shown this NGN3 positive endocrine progenitor cell further differentiate another type of endocrine cell, which in this case, the delta cell and PP cell. In their research, they conclude one model where they hypothesize this, the ARCs and POX4 two transcription factors, actually the downstream transcription factors of NGN3, and they express in the same endocrine progenitors. And they mutually inhibit each other's activation by binding their promoter site. In this case, if ARCs inhibit POX4, the cells commit to the alpha cell fate. If the POX4 win this battle, cells committed to the beta cell fate. But this all proposed mechanism and everything, it's mostly based on the mRNA expression and mRNA level experiments, and also the static endpoint analysis. What we want to see this lineage decision in dynamic manner, in a time-resolved dynamic manner, and of course in a human model. Therefore, we generate the double reporter system by labeling ARCs with CFP and POX4 with mCherry. Then we differentiate our double knock-in reporter system using stepwise differentiation protocols. And today's talk, I mostly am interested in the pancreatic progenitor stage and endocrine progenitor and maturation stage, which I call S4, S5, and S6. First, we want to understand the expression dynamics of this two transcription factor using our reporter system. What we see, these two ARCs and POX4 both start to express in the pancreatic progenitor stage, but we see the difference in their expression. What we see, POX4 indicates the red bar graph, so it picks the endocrine progenitor stage we call S5. And when the cells enter the maturation stage S6, POX4 is downregulated, meaning POX4 is transiently expressed. So on the other hand, ARCs has a delayed expression, and it picked in the S6 stage or maturation stage, and it stays on. If I summarize all things in the graph, there maybe you can see better. The POX4 has a transient expression and peak in the endocrine progenitor stage. On the other hand, ARCs peak later, and it remains, and it has a stable expression pattern. study the expression pattern. So next, we want to understand the mechanism behind this lineage segregation, and we take advantage of the recent multiomics analysis, which is combining of the single-cell RNA-seq with single-nuclei ATAC-seq. Since we have a reporter system, we are able to enrich the specific population. What we did, enrich the ARCS, PAX4 double positive fraction, and combine with the unsorted sample. There we have our beautiful UMaps showing the entire differentiation, starting with the non-endocrine cell and pre-endocrine and endocrine progenitors. They are mostly comes from the unsorted sample. And our enrichment yield the beta cell lineage here and alpha and epsilon cell lineage. This is one of the advantages of having the double reporter line, so we can enrich the specific population. There we can also, here is the, aside from the alpha and beta lineage, we are able to capture the epsilon lineage, which is the rarest, the endocrine cell type. Next, we want to compare the ARCS and PAX4 expression pattern combined with the NGN and compare with the NGN3 expression pattern. What we see, the neurogenin 3 and PAX4 almost exactly co-localized, and they are mostly marks the early endocrine progenitors, beta progenitor or beta cells. On the other hand, ARCS mark late endocrine progenitors, alpha cell or polyhormonal cell. They express in distinct region. So we also check we confirm also the enrichment strategy, the accuracy of the enrichment strategy. So we see the PAX4 embedding density and mostly enrich the beta progenitor or beta cell. ARCS enrich the alpha cells or polyhormonal cell. What surprised us here, the ARCS PAX4 double positive fraction, it's also here enrich the alpha cell progenitors. There we check the ARCS and PAX4 mRNA expression. What we see, the reporter double positive fraction has the ARCS mRNA, but it already has a reduced PAX4. This is the first data that suggests that ARCS inhibit PAX4 in ARCS PAX4 double positive cell, but PAX4 does not. It is the first difference that we see in the human endocrinogenesis, but not, we see in the human endocrinogenesis. Compared to the mouse endocrinogenesis suggests the cross inhibition, which we don't see in the human endocrinogenesis. So since it's a multi-omics and mRNA-based analysis, we want to confirm our findings with checking the differentiation potency of this enriched fraction. What we did, we again sort this, the ARCS PAX4 and ARCS PAX4 double positive cell and continue the differentiation in maturation stage and check their fate by checking insulin and glucagon. So the PAX4 enriched cells committed to beta cell express mostly insulin, and ARCS enriched cells committed to alpha cell mostly expressing glucagon. In alignment with our multi-omics analysis, we also see the ARCS PAX4 reporter double positive fraction also commit to the alpha cell by mostly expressing glucagon. So this is the single cell RNA-seq part. So we also have the ATAC-seq. Then we are able to check the ARCS and PAX4 locus, whether they directly binds or not. So it is the estimation-based, correlation-based system. But what we see, there is no PAX4 binding site of the ARCS locus. And also there is no ARCS binding site of the PAX4 locus, which suggests they don't interact directly. So Interestingly, what we also see ARCS binding site in ARCS locus, suggesting the ARCS may regulate itself. In this, our single cell, single nuclear ataxic also enable us to map the first detailed gene regulatory network combining both ARCS and POX4. As you can see, it's also there are many interplayers, many transcription factors in between, and again suggest they do not directly interact. We also found many known or novel upstream and downstream targeting factors of ARCS and POX4. And we want to check the accuracy of this gene regulatory network. There we interest the FOXO1, which is a direct downstream targeting factor of ARCS, and it is mostly enriched in the alpha progenitors. We first check the FOXO1 inhibition pattern. It's mostly co-localized with the ARCS. There is a FOXO1 inhibitor commercially available. Therefore, we test our system. We inhibit the FOXO1 using inhibitors, which yield the increased number of the POX4 positive cells. With the potency assay and the multiomics analysis, now we are suggesting a new model to explain human alpha versus beta cell fate decision. In the mouse model, which they shown to the cross inhibition define the phase segregation, it's unlikely to happening in the human endocrinogenesis. What we see, the POX4 expression follows NGN3, and these NGN3 POX4 double positive progenitor further than can differentiate the beta cell or alpha cell. The cells never sees ARCS can commit to the beta cell fate, but if the ARCS expression is ARCS expressed, and it indirectly suppress the POX4 and it commit to the alpha cell fate. So, and the last part of my talk, since we have a beautiful system to dynamically visualize alpha and beta lineage decision, so we want to test our system, and we want to just answer one of the obvious questions. Can we manipulate this lineage decision? We have a list, but we narrowed down one or two, and today I'd like to show you one of them. It's an artemether. It's an anti-malaria drug. It is found in the drug screening of ARCS overexpressed cells as a potential of the ARCS inhibitor. It is found in the Kubijek lab, and the same lab later they shown artemether treatment in human alpha cell line result in reduced ARCS and glucagon expression and leads to alpha to beta transdifferentiation by inducing POX4. But I must say this artemether, it's a very controversial topic, and different lab yield controversial results. So the Husing lab use the artemether, and in human islets, I think they found like artemether actually did not cause the transdifferentiation. It leads to dedifferentiation by reducing all alpha and beta cell marker. Kastner lab use a different approach, and they use not artemether but one of the derivative of the artemether, artesunate, and they treat the mice with artesunate, then which they don't see significant difference. So the artemether test in human islet, mouse, adult human mouse islet, human islet, human cell line, and zebrafish, but never tested in the in vitro human model. Therefore, it was appealing for us. Then we test our system. We add artemether during pancreatic progenitor and endocrine progenitor stage. We also perform multi-omics to reveal this mechanism. What we see, the artemether really reduced the non-endocrine cells, and it induced the pre-endocrine, endocrine, and alpha and beta endocrine cells. as if it accelerates the differentiation. We confirm our findings in multiomics with the staining. What we see here, artemether-induced pancreatic progenitor marker significantly, and also the endocrine progenitor marker significantly. But what was the striking result is here, the artemether actually induced the POX4 positive cell number 30% to 90%, and in endocrine progenitor stage. And it then yields to the increased number of the insulin producing cell, and at the same time, it reduced the glucagon positive cells. So in the end, we generate the double reporter IPS line, which enable us to do the temporal analysis of the alpha versus beta sulfate decision and consequence of the drug perturbation on alpha and beta sulfate decision. And with our multiomics potency assay, and I didn't show you, but here with also the proteomics show that suggest a new model, human-specific model for alpha versus beta sulfate allocation. And we are able to generate first detailed gene regulatory network involving ARCs and POX4 and showing the downstream and upstream targeting factor of them. We also test the FOXO1 inhibitor in vitro differentiation, which then yields the the commitment of the beta cell by increasing beta progenitors, in our case, POX4 positive cells. Lastly, we show in the both multiomics and also with our stainings show that artemether increased beta cell differentiation at the expense of the alpha cell differentiation in our in vitro differentiation. So then we bring a new data in artemether controversy. So outlook, how can we use this line in future? One of them would be like we have a limited control of the proportion of the beta cell and alpha cell that we generate in the islet. And along with, we also generate a substantial amount of the unwanted byproduct cell. So this double reporter system may help to improve the islet composition. And if you're interested in this topic, also one of my talented colleagues and also friends, Nike, has an amazing talk and would be on this Thursday. Don't miss out. And also our system is a unique tool to study human-specific developmental process. So in a time-resolved manner, not the static endpoint analysis. Therefore, it's kind of leverage our understanding of the human endocrinogenesis. Lastly, so this double reporter system is a really easy readout to us and offer the advantage of for the drug testing, as well as the identifying the novel compounds and without interspecies difference. We know we have many chemicals showing potent result in the mouse, but not in human. It sometimes fail in the human clinical trials. So using in vitro human model maybe eliminate this problem. And also, since we are using the human model, it would be more efficient to test the efficacy or toxicity. If you want to read or learn more about it, so we put our paper in bioRxiv now. If you'd like to check out, and if you have any suggestion or question, you can always ask me. With that, I'd like to thank you for inviting me. And also, I would like to thank the whole team, talented, amazing scientists I've been working on. Of course, my supervisor, Heiko and Ingo, to give me such exciting project. But big thanks goes to the stem cell team. They are amazing, and it was always a privilege to working with them. So thank you, and I'm happy to take questions.
Thank you very much, Melis. This is a beautiful system that you've basically brought to life and then just sort of walked everyone through. I'll open it up to questions. I see that there's several stem cell experts in the audience. So if you'd like to ask a question, please just unmute yourself or raise your hand. Here's a question from Dr. Julia Panzer, coming from the West Coast, California.
Yeah, hi, thanks. Thank you for the beautiful talk and congratulations on submitting your thesis.
Thank you.
I hope your defense goes well too.
Hope so.
I have a question and I'm not sure, maybe I got it wrong, but from in the very beginning, you showed the expression patterns of ARCs and PAX4, right?
Yeah.
And PAX4 comes before ARCs. Is that correct?
It's a bit tricky, but it's not easy to see in the live flow cytometry. But if I combine the flow cytometry analysis with our multiomics, yes, PAX4 comes earlier than ARCs. At least the mRNA expression, yes.
So
But
that would mean that
on the same time.
So that would technically mean that beta cells are formed before alpha cells, which is in development completely the opposite, because usually alpha cells are formed a few days prior to that. And there's the hypothesis that alpha cells serve as guidance and release paracrine signals for beta cells to start functioning properly.
But this is in mouse, right? In the mouse, alpha cell comes
Wow.
first. But a human, it's now it's also it's a bit under debate. So the beta cell comes first in human, right? In human endocrinogenesis.
Wow.
So yeah, as far as I know. But in our system and the time point that we did multiomics actually, so we see PAX4, it's very early beta cell marker and also the endocrine progenitor marker. So technically I would say yes, but yeah.
But did you ever look at function of your cells, like how they function, especially the ones that the alpha cells that have both PAX and ARCs expression?
Function meaning like dynamic insulin secretion or glucagon secretion.
Yeah, probably
Yeah.
rather imaging, because you want to look at the individual cells that have both markers, right? Versus
Yeah.
the ones that don't. Because I think maybe the timing might be very important for their proper function, because we know that, you know, all the paracrine signals that are released influence the cells and their maturation. So
Well, the potency assay that we perform, when we isolate them, then we put them in the maturation stage. So they should be mature and expressing glucagon and insulin at that level. But I haven't checked the glucagon secretion and under the low and high glucose. I didn't check. It was in my list, but I haven't checked the functionality yet.
thank you.
I wanted to ask a question, you know, with the whole, when you're talking about the easy readout of the alpha beta lineage decisions, you know, how great that is for drug testing. Can you maybe use or share a specific example, Melis, where that might, you know, just sort of like right now could be useful?
For sure, it's artimeter.
Yes.
It really induced the endocrine induction as well as the increased beta cell commitment in our system. But in a differentiation protocol, we add many cytokines and chemicals. I am able to check every single cytokine, whether it drives the ARCs, whether it drives to the ARCs expression or PAX4, or some chemicals or some drugs. So I've been testing some agonist, GABA agonist, antagonist, I would say, or some Wnt inhibitor or so on. There I can see difference, I would say. Maybe I can't say that much. But yeah, to me, it's easy to, I just take out some cell and put in the flow of cytometry, and it took me 5 days, in 5 seconds, or more than under a minute. Or I can take out some aggregates and put in the confocal, I can see the reporter expression. So whether it's increase or decrease. That's why I said it's an easy readout to me. There I see we have some we see some chemical, it's strongly induce and favor to alpha cell or beta cell fate. Artemeter is just one of them.
Mm-hmm. Great. Yeah, I just wanted to sort of unpack that a little bit. I see some big names here. Actually, Jeff Millman, I didn't know if he had anything to ask. He usually has some very good questions.
Okay.
Not sure he's able to. But yeah, I think this is a really interesting system. You've done a great amount of work. I wonder, in terms of, next step of dissecting out the other islet cells, what are your thoughts on that? Would you apply this same meticulous approach, or can you kind of like use what you've already have as a stepping stone to next level these and understand these other islet cells? Guess I
Yeah,
would
well,
like to
this is
ask
a
that.
really, really good and interesting question, and it's been bothering me a lot also. So we see the epsilon cell, and I think we are the only lab showing the epsilon progenitors and epsilon cells in vitro differentiation, just because of our enrichment system, because the epsilon lineage also close by the alpha lineage decision. Since we enrich the arcs, there we are able to capture two. So we have some transcription factors suggest to govern this epsilon lineage too. But it's yeah, and the further study to investigate this part would be amazing. Because in our field, we are more focused on the beta cell due to diabetes. And also we have 2-3 differentiation protocol to generate the alpha cell, right? But we don't have a protocol for generating somatostatin expressing the delta cells or other endocrine cell type. It would be amazing to have a
Yes.
protocol for them and combine to have actual islet, like human islet. That would be amazing. But yeah.
Yeah, for sure. And I mean, I think that people are really interested right now in enriching the delta cell as well. So I think that would be
Yeah.
very helpful.
It
Just
is
have-
also a hot topic.
yeah.
One question.
Oh, yes.
So, very nice talk, Melis.
Thank you, sir.
I wanted to know, like, how did you decide on when to add Artemis
Artemeter.
artemeter, and do you add N-acetylcysteine protocol? Because I see like in the Balbo protocol, they add in the S7 stage, and I see
Yeah.
like completely the beta cell is one chunk and the alpha cell is totally different chunk. Do you see such segregation in yours, or is it just
I don't.
juxtaposed? Okay.
No, I don't see this segregation, but yeah. So maybe I must like add a few things. So the artemeters, since it's an anti-malaria drug, it's a mechanism of action kind of described in the prokaryotes, but not in eukaryote. We don't know the mechanism of action. So in the treat the anti-malaria, they usually combine the artemeter with N-acetylcysteine to cancel its side effect. So I didn't put in here, but I test the N-acetylcysteine. I must say it's kind of partially cancel the artemeter's effect. And it also increase more insulin producing beta cell number, like it's also proposed in the Balboa paper and the recent beta cell protocols, like they are adding in the last stage for the maturation to have more and mature. But my question, the kind of the answer is the lineage decision in endocrine progenitor stage, right? I didn't go further the maturation stage. And the first question that you asked, how did I decide? Adding artemeter. So I had many experiments. I add different stage, combining 2 stage and separately all together. And this is the result that that's a 90% POX4 positive cells I reach adding in the pancreatic progenitor and endocrine progenitor stage, which totally makes sense to me because the ARCs and POX4 switch on the pancreatic progenitor stage. So I just mimic the expression dynamics and add the artemeters. Then I yield this result.
Yeah, it would be good to even have a dynamic GSIS on the glucagon to
I
see the
know this
function.
is your expertise also. Yeah, this is also in my plan. I'm now doing it. I have some cells.
I sense a
Yeah,
collaboration.
congratulations.
I sense a collaboration opportunity here. That's great. Last but not least, Melis, where
Thank
do
you.
you have a postdoc set up for your next step?
Actually, yes.
All right.
I have a scholarship given from the Ministry of Education in Turkey. So in exchanges, I have a position one of the university in Turkey, actually the Ankara University in Stem Cell Institute. So after finishing my PhD, I'm going back to the Turkey and starting my new position in the Stem Cell Institute.
Congratulations. Will you still be focusing on beta cells or
For
type?
sure, this is my passion.
Great. That is really great to hear that, someone who's been so well educated in this field and obviously is very adept and agile in generating a bunch of novel new data in the field will continue to work in type 1 diabetes. Thank you so much for this great presentation.
Thank you
Congratulations.
for the invitation.
Congratulations on your work and your soon, I'm sure, soon to be achieved doctoral degree. Have a
Thank you.
great rest of your day.
Thanks. Have a nice day.